So here's how people use mouse models to test vaccine candidates. So basically, broadly, a major goal of HIV vaccine development is to induce broadly neutralizing antibodies. So these antibodies, they evolve from unmutated common ancestors that go through a very extensive process of affinity maturation. So the question is, how can you design an immunogen that can realize this process? And so to model this in mouse, so basically we express these bnAb precursors, unmutated common ancestor intermediates, and then you can use these mice to test your immunogen to see whether your immunogen can activate the B cells expressing these precursors and mature them toward bnAbs. And generally, I categorize mouse model into two types. And so I'll discuss the pros and cons of testing immunogens. So the first type of mouse model is pretty simple, basically. So based on, let's say, longitudinal studies, you know a particular bnAb lineage come from an unmutated common ancestor, goes through various intermediates. So you knock in these variable regions that encode these UCAs or IAs into the mouse JH or J kappa region. So in this case, because of eddy exclusion, so you express a pre-rearranged variable region of this antibody, and so they'll inhibit the rearrangement and expression of the endogenous mouse antibodies. So in this case, basically, in theory, these mice, they will have a monoclonal B cell population. They will express your target antibody. Then you can immunize these mice to see whether the immunogen can activate these B cells. But technically, actually, it's not that straightforward. And a major problem has been that when you try to express these bnAb precursors, a lot of these B cells, they get deleted, or they're anergic, meaning they don't respond very well to immunogens. So here is an example. So, so when you knock out this variable region exon of a bnAb, let's say heavy chain, into one of the IgH allele. So let's say if you use F1 ESL F1 mouse, the two IgH alleles can be distinguished by polymorphism. So you knock in the variable region of the bnAb into the A allele, and the B allele is completely mouse. So here's a wild-type F1 mouse where you have equal representation of A and B allotype B cells. So here's when knocking innocuous mouse antibody where there's no negative selection. So as I said, because of the exclusion, all the B cells express the allele where you have the knock-in mouse heavy chain. So it's all A allotype B cells. So here is an example of knock-in mouse for VRC for bnAb. So in this case, VRC26 is a bnAb against the V1, V2 region, which is at the apex of the envelope. So in the young mouse, 7 weeks, you already see most of the B cells express the B allotype mouse antibody. So very different from the knock-in of a mouse antibody. So you have a little bit of B cells expressing the A allotype antibody, which come from the knock-in heavy chain. But you can see their Ig level is very low. So usually these B cells, they're anergic. They don't respond well to immunization. When the mice gets older, basically all you have is mouse antibodies. There's hardly any human antibody. So here is pretty much the worst example. Most of the knock-in mice expressing bnAbs, they're somewhere between this. But most of them have some problem. So we've been trying to figure out why you have this problem. How can you solve it? And so we found several reasons, potential reasons. We haven't proven that they're the real cause. One problem is that a lot of these bnAb heavy chains, they don't pair well with mouse surrogate light chains. So during B cell maturation, so you first express the heavy chain. They have to pair with surrogate light chain so that they can progress to pre-B cells where they rearrange express light chain. Then it become immature B cells where they put on the antibody on the surface. So if you have a problem here, then they don't go on further. So here's an example, for example, again, using VRC26 as an extreme example. And so we use this assay basically to test the pairing of the heavy chain with surrogate light chain. So heavy chain, if you transfect by itself into 293 T cells, for example, they cannot fold properly without the light chain, so they cannot get secreted. But if you provide with surrogate light chain, they'll pair, they get secreted in the supernatant. Then you measure the antibody concentration in the supernatant. So that gives you an indication as to how well they pair. So here's when we express the red curve is when we express the mouse antibody, control antibody with mouse surrogate light chain, it gets lots of the heavy chain antibody secretion into the supernatant. If you co-transfect VRC26 with heavy chain with mouse surrogate light chain, so they're right here. There's hardly anything. But if you co-transfect the heavy chain with human surrogate light chain, they seem to pair relatively well. So here's if you express VRC26, the blue curve, with human surrogate light chain. So now you can detect a lot of antibody secretion. So that means a lot of these, at least based on this in vitro assay, if you express bnAb heavy chain with mouse surrogate light chain, they don't pair well. But if you use human surrogate light chain, and they can associate, and so that provides us with a potential way of solving this problem. That is, if you replace the mouse surrogate light chain in the knock-in mice with human surrogate light chain, maybe you can solve this problem or reduce it to some extent. So that's one thing we're testing. Another problem we find is that a lot of these bnAbs, they use human lambda light chain. But in mouse, lambda light chain is rarely used. So in humans, 40% of B cells use lambda, whereas in mouse, only about 5%. And the diversity is very low in mouse. And so in knock-in mice, usually you knock in the lambda variable region, whether they're kappa or lambda, you knock into the mouse kappa lotus, you end up getting a hybrid light chain. So the variable region is human lambda, the constant region is mouse kappa. We find that for some human lambda chain, they don't pair well with their heavy chain in this hybrid context. And so to solve this problem, we're testing this idea that what if you just replace the mouse kappa constant region with human lambda, and then express the lambda variable region in the context of, in association with the lambda constant region. Maybe they can pair better, and then they can get expressed better. And so that's the second thing we're testing. The third potential problem is that these knock-in mice, we think their B-cell maturation process is not entirely normal. And so that's the typical B-cell maturation process. You start with pre-pro B cells, and then go to pro B cells where you have heavy chain rearrangement. And then you get to pre-B cells, you have light chain rearrangement. Then they get to immature and mature B cells, you have surface IgM. So a key marker between this, the pre-pro B cell and pro B cell, is that CD19. So at this stage, they're CD19 negative. When you get to pro, they become CD19 positive. So usually the transition is very rapid. You don't see any intermediates here. And so you can see that's pre-pro B cell, so that's a normal wild-type mouse. And that's IgM, that's CD19. Pre-pro B cell, they go to pro B and pre-B. So they're CD19 positive, IgM negative. Then they become immature and mature B cell, they're IgM positive. If you look at knock-in mice, their bone marrow B cell profile is completely different. Even with knock-in of a mouse antibody that I showed earlier, that they have apparent normal B cells in the periphery, their bone marrow profile is very different from a regular wild-type mouse. You have pre-pro B cell, then you can see hardly any pro and pre-B cell. They sort of jump from here to immature and mature B cell, presumably because you already gave them a pre-rearranged heavy and light chain. They don't go through the pro and pre-B cell stage. If you look at the VRC26 knock-in mouse, where hardly any of the B cells go to the periphery, you can see they pretty much get stuck at this very abnormal B cell population. So their CD19 variable, their IgM low. And so you normally don't see this in the wild-type bone marrow. Here's what we think is going on. So normally you go through this process, but in knock-in mice, because you give them pre-rearranged heavy chain, they don't go through this process. As the pre-pro B cell, they start to turn on the expression of heavy and light chain, the pre-rearranged, they jump to immature B cells. But these immature B cells, they're, in some cases, they're abnormal. So that's why you're seeing, so this, they're turning on their CD19, but they're expressing this heavy and light chain in the wrong time. And we think that could be one reason that these B cells also don't develop normally. So to solve this problem, we're using this rearrangement mechanism. Instead of giving them pre-rearranged heavy and light chain, we separate the variable region into gene segments so that they have to go through the normal pro-B cell process. Maybe they'll develop normally. And the other problem is that a lot of these bnAbs, they're autoreactive or polyreactive. They stick to a lot of different things. And that can cause developmental arrest in the immature B cell stage. So here's an example of VRC26 staining to splenic B cells and bone marrow cells. Basically, we label these antibodies with fluorophore and stain them for splenocytes and bone marrow cells. The blue is staining for VRC26. The red is staining for control mouse antibody, which doesn't interfere with B cell maturation. You can see there are a lot more staining of VRC26 to splenic B cells and bone marrow cells. We don't know exactly what they're binding to, but this assay is mostly meant to say that these antibodies are sticky for some reason, and that can cause developmental arrest at the immature B cell stage. So to solve this, we're using this conditional expression strategy. And so here we knock in two variable regions. And so this variable region, which is expressed in B cell precursors, we call them driver because they drive B cell maturation. So for this, we use an innocuous mouse antibody, which doesn't cause a problem in B cell maturation. They're flanked by two log P sites. When the mouse B cells mature into the periphery, they turn on CD21 Cre, which is expressed in mature B cells. That will delete this driver. Then you turn on, you move this bnAb variable region exon for expression in mature B cells. Basically, that will allow you to bypass any checkpoints in the bone marrow. And with this mechanism, we can express VRC26 in mature B cells in the periphery. So that's what I showed before. If you express in the bone marrow, they get deleted. But if you let them bypass bone marrow, then they go to the periphery. And so if you use, so another problem, so that's what I've been talking about is using this knock-in to express unmutated common ancestor. So there's a second problem. Even if you don't have any of these problems, if you use the method to express intermediate antibodies, and that can still cause a non-physiological situation to test immunogen. Because intermediate antibodies or affinity maturation intermediates, they're usually expressed in germinal center B cells and memory B cells. So that's the physiological target for your boost immunogen. But here you're expressing them through B cell precursor. So you're fairly, sort of unfairly targeting them for tolerance control deletion in the bone marrow. And also, even if they get through to the mature B cell, they're expressed as naive B cells. Normally in intermediate, they're not expressed in naive B cells. They respond as germinal centers of memory B cells. Memory B cells, they don't respond the same way as naive B cells. And to solve that problem, so we're adapting, again, the conditional expression strategy and to express intermediate antibodies in germinal center B cells or memory B cells. So the same conditional cassette, except we couple them with AID Cre, which is expressed during B cell activation. And so here is an example. For example, we're using this AID Cre using a YIP indicator. So YIP normally is prevented from expression by this flock stop cassette. If you have Cre, then the YIP turn on. So that's a way of marking where the Cre is active. So here you can see that in germinal center B cells, you have YIP positive cells. In non-germinal center B cells, there's hardly any. So we're using this system to express intermediate antibody in germinal center B cells as a way of testing boost immunogen. So here's a summary of this type of knock-in mouse model. And so how we're trying to improve it. So we're replacing mouse surrogate light chain with human surrogate light chain and expressing bnAb lambda light chain in the context of human lambda constant region. And we're using de novo assembly of variable region exon and so that they have normal maturation pathway. We're using conditional expression of bnAb UCA in mature B cells to bypass tolerance checkpoints in bone marrow, and we're expressing intermediate antibodies in germinal center B cells. So let's say you solve all these technical problems, you get the perfect knock-in mice that they have lots of B cells. The question, what can you learn from this kind of mice? So really, these mice are intended to test whether an immunogen can activate the B cells expressing the UCA or intermediate antibody and mature them toward bnAb. So the advantage, of course, you have an easy result. Easy readout, you have lots of B cells there. It can give you a sort of yes or no answer to see whether immunogen work at all. And these are, of course, the obvious limitations. It's very contrived. You have lots of B cell precursors that you normally don't get under physiological conditions. You don't have competing antibodies. That's a major problem in immunization. The immunization go off track. And so you're knocking a fixed antibody with a fixed CDR3. And the CDR3 is extremely diverse in humans or any immune system. And in a real human public immunization situation, a lot of people may not even have that CDR3. So this really is just to see whether immunogen can work at all. Of course, if it doesn't work, then there's no point in going further. So you have to go back to the drawing board. So the second type of model is a bit more physiological. And so basically, we put in the gene segments instead of putting a fixed rearranged variable region. We'll put in gene segments so that they can rearrange de novo. You have diverse CDR3, and also you also have a lot of mouse antibodies in this mouse model, so that they serve as competition to see whether immunity is specific enough. So we previously used this model, mouse model, to test to generate diverse precursor of VRC01 bnAb, and so we use that to test a sequential immunization scheme to see whether they can mature this diverse VRC01-like precursors into neutralizing antibodies. So we're now expanding this approach to generate diverse bnAb precursors for major bnAb epitopes. If you look at bnAbs that targeting major epitopes, they tend to use some common gene segments. A lot of them use these 3-3 segments, presumably because it's very long. A lot of bnAbs have long CDR3s. And they tend to use human JH6. So these are the longest D and J segments. And also for V segments, for a particular epitope, they tend to use some common V segments, be it 3-30 or 33 for V1, V2 antibody, for example. So we're knocking in these gene segments to let them rearrange so they can you can generate diverse repertoires for precursors for particular for antibodies for particular epitopes. So if you get this kind of mouse model, so what would be the result? So these kind of models can be used to test whether your immunogen is selective for bnAb epitope. So you have competing antibodies in this case. And also you have diverse CDR3s. So you can also ask whether immunogen can mobilize diverse precursors with related CDR3s. And so it's a little more physiological relative to the first kind of mouse model. And but still there are limitations. So no matter how you try to make it diverse, the precursor frequency at CDR3 distribution will still be different from a real person. And the competing antibody would be mouse rather than human. And of course, we're not doing anything for the T cell arm. So the MHC and T cell repertoire is completely different. So no matter what you do, well, a mouse cannot substitute for a human. So I guess if your immunogen is promising enough in this kind of mouse model, it's probably time to move into NHP or clinical trial. So I'd like to thank the many people who contributed to this work I talk about and the funding sources. Thank you.